The two types of cell division are generally called mitosis and meiosis. But, strictly, these terms refer to the stages of division of the cell nucleus for somatic, non-reproductive, and reproductive cells, respectively. Mitosis. Mitosis is the process of cell division. A cell nucleus divides to produce two nuclei. All the activities in the cell are controlled by instructions which come from the nucleus. These instructions are coded into a molecule called deoxyribonucleic. It is necessary that every cell has a copy of every piece of DNA from its parent cell. Each of the daughter nuclei contain the same number of chromosomes and have the same genetic makeup as the original parent cell. The contents of cell and organelles are equally distributed between each of the cells. Mitosis. Mitosis is a continuous cycle of events. The phases of mitosis are not all of equal length. Interphase takes up 90% of the cell cycle time. Prophase, metaphase, anaphase, and telophase all happen quite rapidly, perhaps in an hour in a typical mammal. Late interphase. The chromosomes are not visible as they are so thin and elongated. In late interphase, the DNA of each chromosome replicates. Prophase. Chromosomes shorten and thicken by condensing their helical structure. The two pairs of centrioles separate two opposite ends of the cell and the fibers of the spindle develop. The nuclei gradually disappear and the nuclear envelope starts to break down. Prometaphase. Nuclear envelope disappeared. The spindle is fully formed with the centriole pairs positioned at the end of the spindle. Each chromosome is seen to consist of a pair of identical sister chromatids. Metaphase. The chromosomes are lined up on the equator of the spindle. The arms of the chromatids wave about, generally in the cytoplasm. Each pair is joined at a centromere. Anaphase. The chromatids start to move away from each other. Eventually the centromeres split. The chromatids appear to be pulled along by their centromeres, which are attached to the spindle fibers. Late anaphase. The cell gets longer and thinner and the chromatids complete their movement. The organelles move so that they are equally spread between the two ends of the cell. Telophase. The new nuclear envelope starts to form around each group of chromosomes. Each nucleus now has a copy of the chromosomal DNA material with all the genetic information of its parent cell. The chromosome uncoil and lengthen and the spindle fibers disintegrate. The cell membrane starts to pull together to form two cells. Importance of mitosis. Replication of genetic material. Growth. Asexual reproduction. Regeneration and cell replacement. Meiosis. Meiosis is the process of cell division which occurs during the formation of eggs and sperms. Meiosis produces cells which have only one copy of each piece of information and only one of each of the pairs of chromosomes. In other words, meiosis produces cells with half the original number of chromosomes, sometimes called as reduction division. Interphase. The DNA is present as thin strands which are not visible with a light microscope. The nucleolus shows up as a dark spot in the nucleus. Two pairs of centrioles can be seen together near the nucleus, which is surrounded by a nuclear envelope. Interphase is sometimes called the resting stage. The cell is not resting. During this phase, the cell is growing, organelles and DNA are being replicated. First meiotic division. Prophase I. The DNA has condensed and is now visible as Discrete chromosomes. Each chromosome consists of sister chromatids, joined together at the centromere. While they are joined, the chromatids are referred as sister chromatids. Prophase I. The centrioles migrate to opposite ends of the cell, and spindle fibers start to develop. Late prophase I. The nuclear envelope breaks down and the nucleus disappears. Homologous chromosomes move so that they lie together in pairs. The resultant pairs are called bivalents. Late prophase I. Homologous chromosomes become attached to each other at one or more points called chiasmata. These hold the chromosomes together mechanically and allow sections of chromosomes to be exchanged. This is called crossing over and is an important contribution to genetic variation. Metaphase I. Chromosomes line up at the equator of the cell. Homologous chromosomes remain associated at chiasmata, but otherwise the chromosomes are arranged randomly. Centromeres become attached to the spindle fibers. Anaphase I. Homologous chromosomes migrate to opposite poles of the cell. This movement seems to be brought about by the spindle fibers, but the exact mechanism is not known. Homologous chromosomes are no longer connected by chiasmata. Telophase I. There are now two groups of chromosomes at the poles of the cell. These uncoil and become surrounded by a nuclear envelope to form two nuclei. The centriole in each new cell replicates in readiness for the second meiotic division. There is now usually a short interphase, although DNA is not replicated at this stage. Second meiotic division. Prophase II. In each of the new cell, the centrioles migrate to opposite ends of the cell and a new spindle apparatus forms at a right angle to that from the first meiotic division. The chromosomes condense and become visible again. Metaphase II. The nuclear envelope breaks down and the chromosomes line up randomly at the equator of the cell. The centromeres are attached to the spindle fibers. It can be seen clearly that each chromosome consists of two sister chromatids. Anaphase II. The centromere splits and the chromatids are pulled to opposite poles of the cell. Telophase II. A cell membrane forms across the equator of the cell. Each group of chromatids becomes surrounded by a nuclear membrane and a nucleolus forms. The chromatids uncoil and return to the interphase state. The spindle disappears.